My name is Derek Aday, and I'm the Dean of the Batten School of Coastal and Marine Sciences and the Director of VIMS, and it's my pleasure to welcome you here tonight. I'm sorry that the Provost can't be with us, but she sends you her greetings and well wishes. The TAC Faculty Lecture Series is relatively new in the scope of William & Mary traditions, but it's quickly become one of the favorites. It began in 2012, and the TAC Lectures represent a unique opportunity for the William & Mary community to benefit from the extraordinary expertise of our faculty. They're made possible through the generous contributions of William & Mary alums Martha Tack and Carl Tack, who are here this evening. Please join me in thanking them for this chance to gather and learn. So I look around the room, it's exciting to see students and staff and faculty and community members all gathered together to hear an expert provide a deep dive into their research. That pun was intended. We're headed into a marvelous dive this evening into the world of salt marshes, and I'm thrilled that our speaker is David Samuel Johnson, class of 1964 associate professor at William & Mary's Batten School and VIMS, who will share stories of survival, adaptation, and evolution in the salt marsh. Professor Johnson is a marine ecologist who studies how global change impacts coastal systems. He's been part of William & Mary and VIMS since 2015, and his research continues to attract international attention. Most recently, he was featured in Science News for his work documenting the migration of fiddler crabs into northern states as a result of climate change. He's also explored parasite-host interactions, the impacts of burrowing crabs on carbon cycling, the effects of the BP oil spill on salt marshes, and more. Professor Johnson regularly advises and mentors students in the Batten School and is an enthusiastic multidisciplinary researcher. As you'll soon see, he is passionate about sharing his knowledge with the public as well as future generation of scientists. So a couple of years ago, we started at VIMS a tradition called First Fridays, which invites people to come and share their expertise across the organization through just a short sort of presentation. And when we started that, I asked David if he would be our first presenter. When I did so, his response was, "I don't do presentations, I do performances." I was relatively new, and I thought, I have no idea what this guy's talking about, but we'll go with it, and we went with it, and it was exactly as promised. So I know you're eager to hear from Professor Johnson. I'll wrap it up with a final thank you to all of the fantastic volunteers and event coordinators who made this possible. Please join me in thanking them. And now let's welcome Professor David Johnson for the 2024 TAC Faculty Lecture. David.
Thank you. All right, thank you. Thank you for having me out today. Thank you all for coming out to see me. Thank you for coming to learn about salt marshes, something I'm very excited to talk about. When I was invited back in the summer by the provost to give a talk, she said, "Hey, I want you to talk about salt marshes." And I was like, "Hey, say less," because I love talking about salt marshes. The only thing I like talking about more than salt marshes are my kids. So you can catch me after the show to talk about those. So this is the coffee bean snail, Melampus bidentatus. Most of you have probably never seen a coffee bean snail, but I guarantee you probably at least half of you have tasted its flesh. Maybe not in the way that you're thinking about. Coffee bean snails live in salt marshes. Salt marshes are these beautiful ecosystems that we're so fortunate to have here in Virginia and all along the coastlines here in the United States. They are what we call these intertidal grasslands. These grasslands that exist between high and low tide. So imagine a Kansas prairie that is oceanfront property. So it's flooded here in Virginia twice a day by the high tides. And so for something like a snail, like this snail, which breathes air, by the way, it's a tough place to live because you're living in a place that is sometimes ocean and sometimes land. So you have to live this amphibious lifestyle. And so to live in a place that is a salt marsh, as beautiful as it is, you have to be tough. You have to be adapted to living in a place that is basically a place of in-betweens. Sometimes land, sometimes ocean. This amphibious lifestyle. Salt marshes, though they have in terms of diversity, low diversity of species, they're highly productive in the species that live there, that thrive. For instance, the coffee bean snail absolutely thrives there. If you were to go out, and I know you're eager to, and you harvest all the coffee bean snails in an acre of salt marsh, and then you took a pin and you took their little boogery bodies out and you laid them on a cookie sheet. And then you took your cookie sheets, because you're going to need more than one cookie sheet, and you put them into an oven, and then you cooked them at 200 degrees Fahrenheit for 2 days until they were as tough as beef jerky. You would have, it's delicious if you're a fish, you would have almost 80 pounds of snail meat. That's like almost 2 bags of dog food. Or I have kids, it's almost 2 6-year-olds. That is a lot of fish food, bird food, crab food. So they're highly productive ecosystems. And you have these animals and plants and microbes that live in an ecosystem that's tough, but they're beautifully adapted to living there. And so today I'm going to talk about some of the animals that are adapted to living in a place that's between the tides. And so the next animal I want to talk about is the mummichog. The mummichog is this fish. This is an adult-sized fish here, so it's a small minnow. Maybe most of you haven't seen it. If you're a fisherman, you might have used it for bait, but you can find it here at College Creek. You can find it at Jamestown. You can find it at Gloucester Point. You can find it at Norfolk. You can find it in Wachapreague. You can find it in Virginia Beach. You can find it down to the Gulf Coast up to Nova Scotia. And in 1973, you could find it in space. In 1973, NASA sent 3 astronauts, 2 mummichogs, and 50 mummichog eggs into outer space as part of the Skylab missions. Now, why did they do that? Well, NASA and the Russians noticed when they first started sending astronauts and cosmonauts into space, they didn't feel so well the first few days that they were there. They were nauseous, they had headaches, they couldn't keep their breakfast down. They were motion sickness. In fact, they call it space sickness. You might have experienced this if you've been on a boat. It's the same thing, seasickness. And so they wanted to try to understand this idea of space sickness in astronauts, and they wanted to use fish as one of their models. And so they chose the mummichog as the first fish to send into space. Why do you send a fish? Well, the reason that you get motion sickness is because you're getting signals crossed between what your eyes are seeing and what's happening in your ears. In your ears, you have hairs and tiny little crystals called ear stones, and that are telling you which way is up and down. So in your ear, it's based on gravity. Your eyes are telling you what you can see. When you're in a place, well, like on a boat, or if you're in space, those signals get crossed and it causes you to feel this motion sickness. So fish have similar structures in their ears to humans, and they're small, and they're something you can easily put on board to try to understand this space sickness. So in 1973, that's why they sent them up. So what happened? So when they sent the 3 astronauts up, same thing happened. First few days, they didn't feel so well. Nauseous, headache, couldn't really eat their food. In fact, they asked to have their day off earlier in their missions so that they could try to recover. What happened to the fish? So the aquarium that they were placed into is basically a big Ziploc bag. Think of a thick one. And that was basically taped to the wall of the cabin. So that's the aquarium that they are in. So how were the fish responding to this microgravity when they were sent into space? They did this. If you bought these fish from the pet store, you'd probably take them back because you think something was wrong with them. They left Earth just fine. But in the first few days in space, they would do this looping behavior. And the astronaut scientists would record this looping behavior, and they draw it out. And they would do these loops. And so if you can imagine a Salvador Dali clock, the melting clocks, and a fish was attached to one of the hands of that clock. And so the clock is spinning at the same time, and it just sort of spins all these directions. It spins this looping behavior as they described it. And so the fish were doing that. But then the astronauts started to feel better. They didn't have the headaches, they didn't have the nausea. They were able to continue with their mission. What happened to the fish? The fish stopped doing the looping behavior, but they started swimming sideways. Okay, so why did they start swimming sideways? They started swimming sideways because they started orienting their back towards the light of the cabin, which makes sense to us and to the fish, because when you lose the signal of gravity to tell you which way is up or down, then you can use light to tell you which way is up and down, because to us and to the fish, the sun never shines from the bottom of the ocean, it shines from the top. And so they were using light as their cue to tell them which way was up and down. And so this was coincidental to the time that the astronauts were also feeling better in space. And so the looping behavior that the fish were experiencing was their version of space sickness. And so it was demonstrating that both the fish and the humans were experiencing similar reactions to space, if in different ways. Okay, so what happened to the eggs? The eggs hatched about 2 weeks later. There were 48 of the 50 eggs that hatched. And what's remarkable is that because when they hatched, they didn't experience that looping behavior. And so what we think happened is they experienced all that space sickness while they were in the egg. So when they hatched, they had their backs swimming towards the light. So why did NASA pick the mummy chog? What does this have to do with salt marshes? The reason that they picked the mummy chog is because it's from the salt marsh. They needed a fish that was tough. And you're not going to find a much tougher fish than the mummy chog. I could take this, not that one, I could take a mummy chog and I could put it on a bucket here on stage, leave a very thin film of water, and then I could come back tomorrow and it'd still be alive. You cannot do that with a goldfish. But a mummy chog can breathe air as long as its gills are still wet. In fact, it will flip over to get the other side wet. Now, why is that important to the mummy chog in the marsh? Let me back up. The reason that they picked the mummy chog was because it's tough and adapted to this intertidal lifestyle, which makes it tough. The reason that this ability to breathe air is important for something like a mummy chog is because, remember, we live in the intertidal. When that water rises and goes up over the grass, that grass is where the food is for the mummy chog. That is a buffet. That is where the coffee bean snails are. That's where the worms are. That's where the spiders are. That's where the fish gets its nutrition. That's where it fattens itself up. And work that I've done with others has shown that when you cut off that buffet, their production, their biomass, their numbers plummet. And so when the tide rises, they go up onto that marsh, and they eat, and they eat, and they eat, right? But, the tide waits for no man and no fish. And so the tide leaves. But sometimes you're dallying, like kids not wanting to put on their shoes, and maybe you're eating a snail here or a spider there, and you get caught at low tide up on the marsh. Okay. For other fish, this would be the end. But for a mummy chog, if you happen to be in a shallow pool with water, or if you have damp grass, and you're able to keep your gills wet, you're able to survive for the next tide that's coming 6, 7, 8 hours later. So they're adapted to this intertidal lifestyle. You have a fish that's conquered water, conquered land, and now conquered space. We attack at low tide. Are you ready? All right, I heard it. I heard it from this side. You're ready. This is a really cool story. Okay, so now that I've talked about a fish that's conquered land, Let's talk about another species that's conquered land, right? We're talking about an ecosystem that's sometimes ocean and sometimes land. So let's talk about a crab that's figured out how to live on, well, I'm going to say dry land, but it's damp land, right, at low tide. These are fiddler crabs. Fiddler crabs are these thumb-sized crabs, charismatic. These are my charismatic megafauna right here, right? These are my dolphins and whales. These guys, these are the male. The males have the giant claw that they wave to attract females and to ward off competitors. Now, one of the reasons that fiddler crabs feed at low tide is because that's where their food, which is algae, it's easy to access that. But the other reason that you feed at low tide is because you're safe from predators, such as fish or other crabs that would eat you at high tide, right? So at high tide you have these ocean predators that can come and eat you. So you wait until low tide so that you are safe from those predators. And so you can see these guys that are eating. The males, you have to use their little claw, but this is a relatively safe area for you to reside. Excuse me. Oh. Like I said, it's a safe area for you to be when you feed at low tide. Guys, did you count all the crabs? Because I see one is missing. There was one in the top left corner. He seems to have disappeared. You didn't see that one, did you? Okay, guys, the first time I saw this, I was like, what in the world is happening? I've been studying salt marshes for 20 years, and never had I ever seen something like this. The reason that fiddler crabs are supposed to feed at low tide is because you're safe from aquatic predators. That is a blue crab, a crab that feeds and lives and reproduces underwater. Let's look at this again. See if you can find the crab. 3, 2, 1. Okay. This is on the eastern shore of Virginia. So I discovered 3 years ago that while doing another study, while we're out there, this is total serendipity, that these crabs, oh nice. Okay, so this is one as he was making his escape. So I have seen blue crabs in the marsh at low tide. I thought they were like the mummichogs, just getting trapped by the low tide and they didn't make it out there. But what they're doing is they're digging pits, hiding in those pits, and ambushing fiddler crabs, which come out at low tide. And they're ambushing them like like crocodiles ambushing wildebeest on the Nile River. It blew my mind to see this because it was 97 degrees Fahrenheit. It was hot as phooey. They were, 70 meters away. It's like if we swam a mile down to hide behind a rock to ambush a fish. It was insane to see this. But what this means ecologically is that this crab is able to take advantage of the marsh 24 hours a day. Because we know that blue crabs come up onto the marsh and feed at high tide, just like the mummichog does. But now the crab is, well, it's been doing it for a while. Now we know the crab can feed at low tide as well. And we know that the babies, these really small juveniles, that salt marshes are really important to them as what we call nursery habitats. So work by Ron Lipschitz and Rochelle Seitz over at VEMS have done a lot of work to demonstrate this. And so we knew this about the little ones. The crabs that I showed you weren't quite market size. They're more like the teenager age crabs there. But those were big enough, right, as you could see, to snag fiddler crabs at low tide. And so what I love about this story is that, again, this is something I've been studying salt marshes for 20 years, and I'm still learning something new. But you have this crab that has adapted to this intertidal lifestyle, so much so that it's learned to live not only in the water, but to take advantage of the food resources at low tide. All right, now this is a cool story. So when you go out to a salt marsh, you see all of that grass. When you're kayaking, when you're fishing. And there's two things the grass wants to do. It wants to photosynthesize, and it wants to reproduce. But what it doesn't want to do is get eaten. And so it invests in things that keep it from getting eaten. Chemicals, what we call anti-herbivory chemicals, things like lignin and cellulose are two of those things. These are the same compounds that make up wood. Obviously this is a grass, so it's still bendy, so it doesn't have as much of those lignins and cellulose. There are other chemicals in there. But it keeps things from eating, herbivores from eating them. In fact, you don't see a lot of deer out there eating them. And deer eat everything. In fact, in salt marshes, most of that grass that fuels the food web, again, we have a very productive food web, is not eaten live. Most of it dies. It has to be broken down by decomposers like fungus and bacteria. And once those break down that material, then it's available for things like the coffee bean snail and other consumers to eat it and use that energy. If you drive along the parkway when you can, or you go out to any of the marshes around here, you'll see something like this. These are periwinkle snails. These guys have figured out a way to eat the grass while it's still alive. And you're like, okay, well, you told me that they don't eat the grass alive. Okay, so they actually don't. What they do is they recruit a decomposer to help them. But to do that, they must first damage the leaf to create dead tissue on the leaf while it's still alive. And how do they do that? Well, they have to use their tongue. So snails have a structure called a radula. And it's this toothy tongue that they use to scrape the leaf with. And so if you could imagine, if you have a cat and you let it lick your face, right, it feels like 120 grit sandpaper. If this snail was the size of your tabby and you let it lick your face, it'd be like a cheese grater, flaying the flesh from your face. So this tongue comes in handy for this snail, and most snails, mind you, in trying to bypass the defenses that the grass has. And so what it does, again, is it damages that leaf tissue and allows a fungus to colonize that damaged tissue. So it's decomposing, and then it doesn't eat the grass itself, it's eating that fungus. We do the same thing. We eat mushrooms instead of like the wood that it grows on. PSA, don't go out in the woods foraging for mushrooms unless you know what you're doing. So the snail is doing the same thing. It's eating the decomposer instead of the grass itself. We eat the decomposers instead of the wood. Same thing. When it comes to the fungus, that we call it fungal farming, so you can think of it this way. Which is, right, so it's solving a problem of how to access this fresh material and not waiting for it to die at the end of the season and go to the ground and then be churned up by the tides and the mud and things like that. It has access to material that very few other consumers have access to. So that's using its head, or its tongue in this case. All right, the case of the zombie shrimp. This is what you all came here for. This is a land shrimp. It's also what we call an amphipod. It's also what we call a marsh hopper. And that's the term I'm going to use. Because you are probably familiar with the beach version of this. If you have ever gone to the beach and picked up algae, kelp, whatever, grass that was on the beach, you picked it up and you saw these little things jumping around out there, those are beach hoppers. This is just the marsh version of that. This is about the size of my pinky nail. And so just to give you an idea of what this looks like. So here's a brief video. They use their butts to jump around. Not graceful, but adorable. It's a good way to use your butt. Now this being able to jump around becomes important to our story in just a minute. Okay, so this is Orchestia grillis, and I expect you to remember this name because there's going to be a quiz later, because we are at the College of William and Mary and the Batten School at VIMS, so you need to learn something. So, if you're having trouble remembering Orchestia grillis, let me help you out. Think of an orchestra. It's a terrible violin, but that's okay. Think of an orchestra on a grill. Hot, 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 hot. Okay, so Orchestia grillis, Orchestia grillis, Orchestia grillis. All right, ready? 1, 2, 3.
Orchestia grillis.
Bam! All right, you all passed. That's awesome. A pluses. All right, so this is Orchestia grillis, because now we have to identify the next species. Oh, that's Orchestia grillis too. Okay, when I first saw this more than 20 years ago, I thought they were two different species, but it turns out they're the same species. And the difference between the one on the left and the one on the right is that the one on the right has a parasite. This is a trematode worm that infects this amphipod, or this marsh hopper. And when it does, several things happen. When it gets infected with this trematode worm, it turns this beautiful pumpkin orange. It also changes the behavior of the marsh hopper. So remember, the beach hoppers like to hide under the seaweed. Well, in the marsh, these marsh hoppers like to hide under the grass. But when it's infected with a parasite, this parasite specifically, it wanders out into the open. Work that we've done shows that it's not drawn to the light, it just sort of wanders out into the open where the light is. And light is this cue that tells you there's danger. But they just wander out there, not aware of any danger, like a drunk into traffic. And it also changes another behavior. Their response to a potential predator. And the potential predator in this case is me. You shouldn't be able to do that. You should not be able to do that because, again, if you pick up on the beach, if you pick up a piece of seaweed, if you're in the marsh and you pick up grass, they're scattering. The reason I don't have a video of me trying to pet one of these when they're uninfected is because you can't get a video of one of those because they run instantly. I'm not that talented of a camera person, but I would never get that close. Now, why does this happen? When it gets infected, does it change its color? Do we see these changes in behavior? Well, this parasite is a puppet master, and it's changing the behavior, and it's changing the color because it needs to get to the next host for this parasite. And the next host is a bird. If my finger was a bird beak, it'd be gone, like that. And if you take a marsh hopper and you take it and you let it wander out from the grass where it's easier to see, and you make it this bright orange, which birds absolutely can see in color, which is basically a flash of neon sign that says, "Eat me," then it's easier for this parasite to get to the next host, in this case, the bird. And what I find fascinating about this story was, again, this is total serendipity, we weren't looking for this, is that you see this manipulation happening in the salt marsh, that it's adapted to living in a place and going through multiple life cycles. But also, as a kid and thinking about marine biology, I never thought, hey, when I study marine biology, I really want to study parasites. Not usually what you think of. This, I think of myself as a marine biologist who has a parasite problem because they keep coming into my life. And I think this is a really fascinating story to illustrate the power of parasites because the predators often get all the glory. But parasites can be just as powerful. Right? So when you think about people who want to be marine biologists, especially when you talk to undergrads, they want to study, right, the charismatic megafauna, the dolphins, the whales, the sharks, and those are cool, absolutely. Parasites are cool in their own rights. If you're a dolphin, whale, or shark, you swim around, you consume things to gain that energy, right? Because we're not autotrophs, we're not photosynthesizers. We can't generate our energy that way. But they have complex nervous systems, they move around a lot. The parasite doesn't necessarily have that complex nervous system and doesn't have to go around gathering all that extra energy. It just uses yours. This is a worm. So when you think of worms, you think of earthworms, but this is a worm that lives in a salt marsh. This worm's about the size of a macaroni noodle, but not as thick. What I love about these guys is that, again, they've adapted to living in this intertidal lifestyle. It has, if you look at the stripes on the tentacles, those are not actually tentacles, those are gills. If we grew our lungs out of the back of our head, and they floated around like balloons. But when the tide comes in, it sets those out because it lives in a mucus mud tube. Sets those out, and it uses those other tentacles to gather food like phytoplankton. These are easily overlooked, right? You need a microscope to look at these. Nobody looks at these but just a handful of people in the world. But I'm fortunate enough to be able to look at them. But these are important in our food web. If you went out and gathered all of these in an acre of salt marsh, we'd have 100 pounds of worm, just from that little tiny worm. So again, highlighting the productivity that we have in our salt marshes. And salt marshes are critical if we just focus even on the food web. So I saw a bumper sticker that said, no marshes, no seafood. And so if we have our worms, we have our snails, we have other crustaceans in there. If you love crab cake sandwiches, if you love striped bass, if you love red drum, if you love junior rails, if you love bird watching, that's a lot of fish, crab, bird food that the marsh provides. And that's just focused on the food web. There's many other services that salt marshes provide for us. And so the next time you're sitting down and you're watching a beautiful sunset, you see the birds fly across, or you're having a crab cake sandwich, or you're out there fishing, thank your local salt marsh. So there's my hero marine biologist is Sylvia Earle. I almost said Sylvia Plath. That's a different hero. And to paraphrase one of her quotes, she says, no ocean, no life, no blue, no green. I would modify that to say that no brown, no green, no coasts, no life to add to the blue. Because when we think about the ocean, we think about the big blue that's out there, and that's absolutely important. But again, we have the Batten School of Coastal and Marine Science, and the coastlines are critically important to a lot of our livelihoods, and there's benefits that we derive from them. And so I've been fortunate enough to work in places like salt marshes, this life between the tides. And so not only are there cool stories that are happening there that we see that is easily overlooked, but they provide a lot of benefits to us as well. So one aspect of my research is to look at the impact of climate change on salt marshes. I was driving on the Parkway, this was 2 years ago, when you could still do it easily, over by Jamestown. It was a beautiful Friday night. And I saw these people with headlamps on the beach, and they were throwing cast nets. And I'd never seen that before. So I went down and asked them what they were catching. And somebody reached in and pulled out this, a shrimp as big as their hand. And I'd never seen shrimp as big as your hand come out of the James River down towards Jamestown like that before. And so it was a mystery to me. So I called up my good friend, Troy Tuckey, who's over at VEMS, and that's who you call when you have a shrimp mystery. So I called him up, I was like, I was like, hey Troy, they're catching shrimp as big as your hand out of the James River. He's like, oh yeah, that's probably white shrimp. I said, white shrimp? They're not supposed to be here. He said, well, they are now. They are now? Boop. They are now. So that was surprising to me. And to understand why that was surprising to me, we need to go back to 1994. We could go back further, but let's go back to 1994. That's when Forrest Gump was #1 at the box office. That's when, and shrimping was big. That's when I graduated high school, and that's when we held phones like this instead of like this. One of the great things about VEMS is that we have long-term data sets. We have all kinds of them, and seagrasses and sharks and nutrients. And we do, Mary Fabrizio and Troy Tuckey over at VEMS do these net surveys. And one of the things you can capture with that are shrimp abundances. And you can see for the longest time there weren't many, but then starting in 2019, we start getting a lot more shrimp coming into Chesapeake Bay. And it was a surprise to me because I knew there was a fishery in North Carolina, and I have family down the Gulf Coast. I went to LSU, so I'm all about like po' boys and things like that. But I had not seen them here in Virginia. And we hadn't seen them in any real numbers until about 2019 onward. And so why is this happening? Well, what's happening is we're experiencing ocean warming. So you've heard about global warming, you've heard about climate change. And so as the atmosphere warms, so too does the ocean. Because if you think of the ocean, the atmosphere and the ocean as two bodies sharing a bed, they share heat. And so as the atmosphere warms, so too does the ocean. And so as the ocean warms, you see species in this hemisphere moving from the south into the north. And so we're seeing more white shrimp here in Chesapeake Bay and in Virginia than we've previously seen before. And this was wild to me, right? So, because when I saw this 2 years ago on that beautiful Friday night, I wasn't just looking at a potential new fishery species for Virginia. I wasn't looking at a potential po' boy. What I was looking at was climate change. And so I have my own story of discovery where species that's where it shouldn't be, or at least I didn't think it should be, back in 2012. So we'll go even further back from my trip to 2 years ago. So I do a lot of work north of Boston by about an hour. And in 2012, somebody said to me, hey, we saw blue crabs up here. And I said, no, you didn't. I've been working up here for a decade. There's no blue crabs up here because, as everybody knows, blue crabs are not north of Cape Cod, Massachusetts, because it is too cold once you get north of Cape Cod, because the waters come down from Canada and it makes it too cold for them up there. So there's no way you saw it. You're a biogeochemist, you don't know what you're talking about. And then somebody else said, hey, I saw one too. So I was like, okay, I gotta check this out. And I went out there, and guess what I found? A blue crab. So I started asking around to my neighbors, and it turns out we started seeing blue crabs in the marshes and other places. They were catching them on the beaches in Nova Scotia. They're catching them in lobster traps in Maine. They're catching them on the beaches in New Hampshire and continuing in Massachusetts. And ever since then, in that past decade or more, we continue to see blue crabs out there. So we have the white shrimp, we have the blue crab story. So these are, to me, the clearest examples of climate change, because it's climate change that you can hold, it's climate change that can pinch the phooey out of you, it's climate change that you can eat. And so there's another climate change story involving, again, our good friend, the fiddler crab. Don't worry, he's not going to get massacred here. He too has expanded his range north of Cape Cod. He's not gone as far as the blue crab. He's not yet in Canada, but he has made it to the middle of Maine, which is really surprising to me because of how cold it is up there. But again, the waters are warming up there enough that it allows these species from the south to break that barrier and go further north. And so one of the questions that you may have is like, okay, well, how do these species travel so far? And how are they getting there? Because one of the things that we see in our research is that in our oceans, these sort of range expansions are happening 10 times faster in our oceans versus in terrestrial ecosystems, like our forests, for instance. So why is that? Well, it has to do, in part, to the life cycle. And I'm going to use the fiddler crab as an example. And this is where you get to use the bubbles. So go ahead and get those ready. But first we have to learn about the life cycle. So the female that lives in the salt marsh, fiddler crabs don't travel very far from their burrows, so they're not walking as adults, past Woods Hole up to Boston. Who's making it farther north? It's the young. She releases her eggs. Those hatch into these larval stages. As you can see, they go through multiple stages here before they get back to what we recognize as fiddler crabs. So what I would like you to do right now is take out your bubbles. Get in. And I would like you to blow those, and we're going to go ahead and blow as many bubbles as you can. I want to see those bubbles fly. And I challenge you to get some of your bubbles up onto this stage. Oh, look at that in the projector. Look at that's amazing. Good job, back row. Do y'all have bubbles? Here. There you go. Okay, so the bubbles here represent the larvae that are floating on the ocean currents. So your seat represents the marsh. Just because you were born in the marsh doesn't mean you return to that same marsh. But as these bubbles, you float on the currents. And if you last long enough and the current is just right, it can take you great distances. If this bubble was strong enough, we could take it outside. Maybe it'd make it to Norfolk in one day. Maybe it'd make it to Richmond. Maybe it'd make it up to DC the next. Maybe up to Boston the next. That's almost as fast as taking I-95. But this is what happens with the larvae, with the larvae here. They float on these currents and they're carried great distances. And that's how these range expansions can happen so much faster in the ocean, not necessarily because of the adults, but because of the larvae. So when these range expansions happen, you can get some really cool ecological and biological effects. So one of the things that we see is a change in body size. This is north of Cape Cod versus south of Cape Cod. So Connecticut versus New Hampshire. Look at the size difference in that. If these guys were in the UFC, they would not fight each other because they would be in totally different size classes, weight classes, right? So the one from the north would completely destroy Connecticut. So So we're seeing these differences, for instance, in size classes. So why is that? Why do we see a difference between the north and the south in terms of body sizes, or this larger body size? Well, one of the hypotheses that we have is that the ones north of Cape Cod in this expanded range have fewer parasites. Because when you move as larvae, you don't care, those usually don't have parasites, and so you can leave your parasites behind. You have fewer parasites south of Cape Cod versus north. And if you have fewer parasites, which we have found in working with my good friend Jeff Shields, a fantastic parasitologist over at VIMS, we have found that they have fewer parasites, and so if you have fewer, you can invest less energy in defending against those parasites, and you can invest more in reproduction and growth. So I would like to end this talk by having a little fun describing our hypothesis about parasites. If you'll just bear with me for a minute. All right, you guys ready? Rock and roll, I'm ready. Well, I gotta wet this whistle. All right, here we go. You only live once, let's do this. YOLO. All right, the water's getting hot. I said the water's getting hot like coffee in the pot. The oceans, like the times, they are a-changing. The animals, my friend, they are rearranging where they're found and where they're not. Lobsters are leaving Long Island, and we're finding blue crabs in Nova Scotia. But don't worry, in the salt marshes, we still got Vegeotoa. Enter the fiddler crab, Manuka pugnax, the little crab who learned to dab. Tenacious and pugnacious, now that may seem harsh, but that's what you've got to be to live in an intertidal salt marsh. Before 2000, you would have found it quite odd to find this little crab north of Cape Cod. But thanks to climate change, he has expanded his range far, far north into the Gulf of Maine. As north the fiddler crab wanders, my mind is left to ponder. If you migrate into the land of the expanded, are your enemies left behind and abandoned? I mean your parasites, your predators, your interspecific competitors. As you can see from this vertical bar plot, that if you live in the range of the expanded, one parasite is all you've got. In the range of the historical, the average is one more than 4, but in the range of the expanded, it is exactly 1, no less and no more. In fact, this one parasite is only found in the expanded. All historical parasites have thus been abandoned. You must forgive my lyrical repetition. I'm not nearly as talented as Eminem or New Edition. And by parasites, I mean your metazoans, your trematodes, your nematodes. You can see where I'm a-going. And it's not to a hip-hop career. Now, sometimes you're subjected to a subject and a predicate that's predicated on meditating on the metadata when the data itself is still in beta, and your results can seem quite premature. But today, I am absolutely sure that if you want to outrun your parasite, the best thing for you to do is pack up your stuff and move. Because climate change can give you enough velocity to escape your enemy's animosity. In fact, New Hampshire could advertise in the latest tourist catalogs that specialize in marine crustacean analogs, a slogan that says, hey, were you aware that we've got fewer parasites than Delaware? But will it always be the case that New Hampshire will be this parasite escape? No, because the enemies may soon arrive, and it will be the end of the fiddler's disease-free honeymoon. In the meantime, what does it mean in the grandest of grand schemes? We see that crabs in the ranges that expand increase to a size that is quite grand. So can it be that if you're relatively disease-free, your growth will expand to make it more grand? But that's just me hypothesizing. We need more experiments about fiddler crab body sizing. For now, you should know that if you have an enemy to flee, use the climate to accelerate your escape velocity. And get yourself to New Hampshire with thee, where it's relatively disease-free. Caroline, Valerie, Mac, and Serena, Emily, Rachel, Andrew, and Manisa, these are David's amazing Saltmarsh heroes, because while my data is often filled with zeros, my lab is not. And our science is so hot that we melted the microphone instead of letting it drop. Thank you. An epilogue, if you will allow me. When you hear the term marine biology, this is an image that you often think of. Somebody swimming in the big blue, a scuba diver. In fact, this is an image that you might imagine being on the cover of National Geographic. I hope from this talk that it expanded your idea of what marine biology could be. It could be muddy. It can be in our coastal zones. If I was to submit a National Geographic cover from the Baton School, this would be my cover. This is an undergraduate from William & Mary. She has since graduated, obviously just into it. And this is what our lab is like. And so she and the next folks you're going to see are really what makes this lab run. And so I'm going to say thank you to them. I'm going to let this run through. Thank you. You can clap for people. Of course. Okay, I won't chew the scenery anymore, but I'll be happy to take any questions that you might have. And we have microphones for you if you have a question. We have a microphone runners. So I think, I apologize, I can't see your hand raised, so I'm going to leave it to the microphone runners to help me with that.
So in the expanded ranges of like fiddler crabs, are you seeing other species that are like native to that area being pushed out? Are you seeing any like new like species interactions with like competition or like new forms of predation, things like that?
Great question. So if I understand your question is, as a species moves in, is it pushing these other species out? Is that your question? Yeah, that's a fantastic question. And so we here at VEMS are on the leading edge of just understanding that. And we haven't seen evidence of that yet because the populations as they move in are still relatively small. And so that's a possibility that that could happen, but we haven't seen that yet with the species that we've tracked. I just want to back up on that question. One thing that we've seen these range expansions for, we've been tracking them in the literature as scientists for 20, 30 years. And so we have tens of thousands of species we have identified, but we really haven't just until now started paying attention to what happens ecologically when those species arrive in these new areas. So that's a great question. It's something that we're looking at.
I was just wondering, as the habitats expand because of the warmer waters, do you ever see that the crabs are leaving the southern parts of their like original habitat? Like, do the waters ever get too warm and then they leave?
Ask that again?
So like as the fiddler crabs, like as they're expanding northward because the water is getting warmer, do you ever see in like the southern area of their original habitat that they leave and then like fill a hole, or like they leave a hole in the...
So like a true shift in the
Yeah.
Okay. So yeah, so I just talked about expansions on the north side. I didn't talk about what happened, like a retraction on the southern side. Is that what you're asking about? Yeah, so that happens with some species. We haven't seen it for the fiddler crab, and that doesn't happen for the blue crab either. So, but some species will, if it gets too warm, that southern limit will be too much for them. But we haven't seen that for the species that we study. So that's a great question. I actually want to follow up on the question that we had before here. I was thinking specifically about crabs, but I want to mention a case study with mangroves. So we're seeing the range expansion of mangroves. So mangroves are subtropical, well, they're tropical species, but we see them in Florida, which is subtropical climate. And we're seeing them move north, and they're starting to displace salt marshes. So we are seeing them outcompete those. I mean, it's a total ecosystem change, right? So you're going from a grassy salt marsh to a mangrove, which is trees. So we are seeing that happen. We haven't seen it with the species that we are studying as animals. Great questions.
Hi, David. Thanks for such an amazing talk. I really had a good time. I was just wondering if you could send another species into space. Which one would it be?
Which one would it be? I kind of want to send mummy chogs back. Which one would it be? So I would probably I would want to send a snail. I wouldn't send the coffee bean snail because he wouldn't, but I would want to send snails because I want to see them go through several generations. I want to see what happens with that. And so I would probably need to send a land snail so that because with the marine snails, they have those larvae that float on the currents. You can't really do that in space. So I think a snail would be fun to do because it's easy to keep, and you could get multiple generations fairly easily. I think that would be a great one. I know with other fish, they've looked at like bone density and things like that. So they've sent other fish up there as well. But that would be my question, is to look what happens when you have multiple generations, right? Because we're trying to send humans, further than the moon. So you imagine there's multiple generations that we're going to be working with there. So, I like that question.
What would happen if you ate one of those orange marsh hoppers?
If I ate one of them? Okay. Would I turn orange? No, I would not. Would I wander out into the light? So I'm not exactly sure what would happen if I ate that one. I have not. So let me say a couple of things. So a couple of things could happen from the point of view of me versus the parasite, right? There's two things happening. If I ate it and it was able to, so it has a little cyst in there, and that was able to, we'll call it hatching, into the adult worm, and then that worm lays eggs. So what happens in the bird is it lays eggs, and that bird poops out the eggs, and then a poop-eating snail comes along and eats the eggs. And then that snail becomes like a Xerox machine pumping out like just different this asexual forms of the parasite that then swims and finds the marsh hopper. Let's say that I am the host that is able to do that. I would be kind of a dead end because I'm not going to be in a position for the snails to actually eat the eggs, right? So, okay. The other thing is The other thing is, I may not be the right host for it. I may be what's called an incompetent host, which is what my friends call me when they come over and I don't give them snacks. Parasite pun. So I'm not the right host for it. So the other thing I would say is, if you ever go on Survivor, don't eat raw stuff. cook it, because you never know. I mean, there could be some pretty nasty stuff out there. So, especially crustaceans like that, and especially snails. Don't eat slugs and snails. Make sure they're cooked. Don't eat them raw. You can eat them cooked, don't eat them raw. So kill those parasites. Great question. Oh, by the way, that question was asked by David Steinberg from VEMS, who's done some fantastic work in the Antarctic, working with krill and all kinds of things with the carbon cycle, and gave an amazing talk for TAC about a decade ago, talking about poop, which was awesome. So I hope that I got somewhere close to the Steinberg standards.
Hi, David. That was a great talk. I had a question. Now, knowing everything you know about marshes and for how long you've been working, if you had to be a species in the marsh, do you have one that you would prefer to be?
Oh, what species would I be in the marsh? Knowing what do I get to keep the knowledge I have?
Yes.
Oh, I get to keep the knowledge I have. Oh, that's an advantage. Okay. I would be a fiddler crab because I would know to stay away from those dastardly blue crabs who've learned to hunt on land, by the way. I would absolutely be a fiddler crab. So that seems like a fun life. Cool questions. Any others?
Hi, thank you for the talk. I had a follow-up question about the space mummy chogs. So how did they, well, first off, what food did they send for the fish? And also, how do you feed a fish in zero gravity? Because I feel like the fish flakes would not make it into the tank.
Yeah, right. So that's a problem in space, is water is a problem in space, right? So in my reading and my request to NASA, I found nothing. What I will tell you from my digging is that the mummy chogs did not make it back to Earth alive. So my assumption is that they may not have been fed, or they might have had you can get those vacation blocks. I don't know. I didn't see anything in the methods about that. It was very limited, and maybe it's lost somewhere in the archives. So, and the astronauts who were on that mission have since passed, so I've not been able to find that information. So after 4 weeks, the two mummy chogs that were already hatched that were sent up had died after 4 weeks, which is still a long time if you hadn't been fed. So it's still a pretty tough fish. So I'm not sure how you would do that. That's an engineering problem. But I'm sure they could figure it out. Anybody else? Well, fantastic, y'all. Well, thank y'all for having me out. This was amazing. Oh, sorry.
Hi, sorry. I was just curious about the anatomy of the monotrochs when they went to space. Did the swim bladders change as well as like the skeletons? Because I know when astronauts go to space, they have to go like through physiotherapy and stuff just to maintain their fitness when they come back to Earth. Like, was there any changes within their like sort of anatomy when they came back? Like, if there's been other missions with other fish when they came back to Earth, like I know they've sent rice fish to space.
Yeah. So yeah, he's asking about were there changes in the physiology other than looking at this behavior. So they didn't look at so they didn't look at those things with the mummy chogs, but they have sent other fish, as you mentioned, like the rice fish up to space, and so but it's only been a handful of fish that they've been able to send up, right? So with the rice fish, they saw a change in bone density. They saw less bone density. The same thing that you can see in humans who've been in space for a long time, because you're not working, right? We don't have gravity pulling down to cause you to build up your bones and your muscles, right, as well. So you see similar things happening with the fish that you would see in the humans. I haven't seen anything about this. I think you were asking about swim bladders, but I know you do see changes in physiology, such as bone density.
Coming out and sharing your evening with us. How about David Johnson?
And don't forget to visit. We have a setup of a marsh. So people from my lab have done a really great job of setting up a marsh right out here. So take a look at it. Thank y'all.